Hello, this is Dr. Mandy, and I wanted to put together this presentation that's been long awaited and needed, as I've spent the last several years really diving deep into oxalates and how they're truly impacting various diseases, not just bladder pain syndrome, but anything that is inflammatory, degenerative, and that's associated with oxidative stress. And I wanted to dive in a little deeper than some of the general content that's out there. This is going to be a relatively long lecture, so feel free to pause whenever you want to and return whenever you can. I am sure you're going to find this very, very enlightening. So what are oxalates? Oxalates are essentially anti-nutrients that are found in plants, and they're not found uniformly in plants. For example, we can see different concentrations in different parts of the plant. The highest concentration are found in foods that are in the goosefoot families, such as spinach, beet, chard, and rhubarb, though we also see them high in nuts and other root vegetables as well. And the theory is that because parts of the plant are producing oxalates in response to some stressors, for example, insect infestation or being nibbled on by rodents, or even drought, there is a variable amount of oxalates even within the same food. You can have one field of spinach that has a high level of oxalates and another that has slightly less level of oxalates, and cooking techniques are also going to influence the level of oxalate. But generally, the highest concentration are found in parts of the plant that the plant needs to live on. So the roots, for example. And then like sweet potatoes and white potatoes, ginger and turmeric are rhizomes, and they also have high levels of oxalate, as well as leaves of spinach, chard, and rhubarb. Now there's generally lower level of oxalates in part of the plant wants another organism or bird to eat, so the fruits. And again, that's theoretically due to the plant needing the organism to eat it to spread the seed. But it's really important to know that oxalates are also produced by yeast and fungus, an area that's not really focused on. And this occurs in molds as a fermentation byproduct. So if we see colonizations of these organisms in our gut, which is rather rampant, we may have a new source of oxalates within our system. And additionally, oxalates are produced, but in small quantities as a byproduct of your metabolism. And there are individuals who have genetic predispositions that cause them to produce excessive levels of oxalic acid, or there can be things upregulating it like nutrient deficiencies or even oxidative stress. And finally, oxalates are mineral chelators, and that's the main mechanism by how they do harm to the body, primarily targeting calcium, magnesium, zinc, and iron. And the affinity for oxalate to calcium is the strongest. That's because oxalates are negatively charged and the minerals are positively charged, so they attract each other. And as a result, we see anywhere from 60 to 80% of kidney stones being calcium oxalate precipitates. So why the focus on oxalates? Well, they have a negative impact on the body through very many mechanisms. These include things such as mechanical shredding and the increase in free radicals and the decrease in antioxidants, which are rampant. And I see this continuously within my clinic. They're also associated with multiple varied pathologies impacting a tremendous amount of different organs and body systems. And they are an independent trigger of the upregulation of the NOX1 enzyme, becoming an independent trigger of mast cell activation syndrome. And this can be caused by multiple environmental causes or dysfunctions in the body and even genetics. So on this slide, you can see various types of oxalate precipitate. And so on one side, you see iron oxalate, and then there's calcium oxalate and sodium oxalate. So just to make you understand this clearly, 60 to 80% of kidney stones are generally calcium oxalate precipitate. So you can see the one in the middle. And they're very inflammatory, but inflammation is such an abstract concept for most people. You may hear people say, my tissue's inflamed, and oh yes, I'm feeling pain, and yes, I'm feeling discomfort. But with oxalates, it's really important that we remember this is a physical mechanism. They're crystalline precipitates that get deposited in your tissue, and we can find them deposited in an extraordinary amount of tissues. And this is not just limited to kidney stones. There have been reports of oxalate precipitate in autopsies of brains, in the eyes, in the lungs, in the heart, in the colon, in the breast. even in breast cancer, and in the kidneys and connective tissue. And it's really important for you to understand these precipitates hijack the entire sulfation system of the body. And I'll get into that a little more. I find it interesting when people on various social media sites and these IC sites argue if oxalates are really an issue. I think the disconnect is that they really don't know what they are, and there are even some nutritionists and dietitians who are still prescribing high oxalate diets as well to people with IC. Truthfully, they are a poison in the body, and they poison us in several different ways. One of the main ways is that they destroy mitochondrial function through several different mechanisms, and they can halt ATP synthesis and can shred intracellular organelles, including the mitochondria, and they alter our redox homeostasis. And these include various mechanisms, including the depletion of glutathione, the depletion of superoxide dismutase, and the depletion of NADPH oxidase, which we need to reduce glutathione from its oxidized to its reduced state. And this happens through the upregulation of a pathway called NOX1 or NADPH oxidase 1. And this upregulation of NOX is pretty significant because it creates additional superoxide, and this is how we get the depletion of superoxide dismutase. And there's actually some people who may have polymorphisms as well in this particular enzyme. Now, if we exceed our capacity to convert superoxide into hydrogen peroxide, we then develop an independent trigger for mast cell activation syndrome. And oxalates can be an independent trigger for people who are struggling with mast cell issues. So let me repeat this. Oxalates can be an independent trigger for people who are struggling with mast cell issues. Part of my mission and others within the community, such as coaches like Emily Gleiber, and even scientists like Susan Owens, and many others within the functional medicine space, such as Monique Attinger, is to get people to start thinking about oxalates as a secondary mycotoxin. And this is a little outside of the norm, both for people in the mold community, but also for people within the oxalate community. When we think about what are mycotoxins, well, they're the toxic byproducts produced from molds. And what is oxalate? Oxalate is the toxic fermentation byproduct produced by colonized molds. So while we will not inhale oxalic acid if we're in a moldy room, if we inhale mold spores, which then take up residence in our system, and they start to colonize in our gut, and they start to ferment different compounds and produce oxalic acid, we now can see the relationship between oxalates and mold. So oxalate hurts the kidneys as well as other tissues such as the brain, the eyes, the heart, the nerves, the muscles, the connective tissue, because it's so insoluble. We're stuck with it once it is aggregated into these precipitates. We're at least stuck with the precipitates, and we're not going to break them back down. They're micron-sized and they're nanocrystals, and they physically shred surrounding tissues where they're deposited, and they can also aggregate into larger stones, and that can allow it to cause more harm in the tissue. And we will see most of that harm when they are deposited, whether it's the brain, the lungs, the blood vessels, the joints, the bones. And this can cause systemic pain and dysfunction. And as I mentioned earlier, this is also exacerbating the inflammatory cycle because we do end up depleting our antioxidants like glutathione and NADPH through the NADPH steal in the upregulation of NADPH oxidase. So once those crystals are deposited into tissue, they mechanically shred the surrounding tissue because they're like little needles or little razor blades or shards of glass in the body. And because they are so small, micron-sized, nano-sized, they're able to enter the cells and they can cause progressive harm as they do. And we can see things like apoptotic and necrotic cell death. We can see the rupture of cell membranes. We can see lysosomal rupture, cell shrinkage, nuclear shrinkage, destruction of intracellular organelles, but this is not limited to just the mitochondrial. So this is part of the connective tissue that we see between chronic fatigue syndrome and hyperoxaluria because of the physical destruction of the mitochondria. You see, it's really hard to make ATP if you don't have good functioning mitochondria. So one of the tricky things about oxalates is they can come from a lot of different sources. we can see genetic predispositions, and there are a number of places we can see this. We'll take a look at those details in a little bit, but I do want to highlight that this is really the minority of people who are dealing with the genetic hyperoxaluria. I usually see about 10, maybe 15% that are genetically expressing those predispositions. So it's possible, but it's definitely the minority. So let's go through some of these. Fat malabsorption, that's a big one. And it's one of the primary mechanisms by which we develop hyperoxaluria. And I would argue probably the most overlooked. So what happens is we're not emulsifying our fats as seen with people who've got bile insufficiency or even low enzyme secretion. So let's just imagine, let's say you have this non-emulsified dietary fat in the digestive system. Let's say you had a really high fat meal and our dietary minerals get bound up with these undigested fats, and the minerals and the dietary fats bind together, and they form these soaps and end up being excreted in the stool. And rather than binding to oxalic acid in the gut, allowing the oxalate to be excreted, they allow passive absorption of the oxalate. So we lose our fats, including our healthy fats, and we end up losing our minerals because we end up with excessive oxalates, and we get this passive absorption through the increased gut permeability, and we get a diffusion of soluble oxalate into the intestinal lumen. So there's this dual mechanism where that's fat malabsorption that can result in hyperoxaluria. And then take it even further, then you're going to see a depletion of your fat-soluble vitamins, and many are your antioxidants, like your vitamin A and your vitamin E, for example. So leaky gut is another issue, and this can cause histamine problems. So similar to the leaky gut that's problematic, in which bile acids can create this, when we have leaky gut, we get this passive absorption of oxalates, and people who are dealing with a lot of histamine issues or poor histamine clearance or overproduction of histamine for a number of different mechanisms do become more vulnerable to overabsorption of oxalates. And we do also have to combine that with a genetic predisposition where some of these histamine genetics, such as the ABP1, which is the DAO enzyme, or HNMT, or even the HDC enzymes. So there is more to the layer than just the leaky gut. And then there is the low sulfate. This is another often missed aspect of hyperoxaluria. So when there's low sulfate as a result of things like maybe there's a SUOX polymorphism, which can impede sulfite to sulfate conversion, or this can allow for an increase in oxalic acid. This is because sulfate and oxalate share solute carriers or transport mechanism into the cell. So when oxalate builds up on one side of the cell membrane, it forces sulfate through that solute carrier across the membrane. So if you have a lot of oxalate in the cell, it forces sulfate out, which ends up excreting or wasting sulfate in the urine, making low sulfate independent mechanism of hyperoxaluria. And I have seen when I run the metabolomics, which uses urine for some of these components, I have seen high levels of sulfate in the urine. And that does give me a clue that there may be some excretion or wasting of sulfate because of oxalates and the presence of oxalate. All right, next is low citrate, and that allows for the crystallization of oxalates, and we can assess this on the Nutravel, metabolomics, or the organic acid. I have to be honest with you, I don't see this very often. Next is dysbiosis. So generally we think about the species of bacteria called Oxalobacter formigenes, or formigenes, however you want to pronounce that. There's several other beneficial bacteria that can potentially degrade oxalates, but they do under very specific physiological conditions. And I can tell you, I've seen it with certain strains of E. coli, and I've also seen it with Enterococcus faecalis, and they're both oxalate degrading. And what I have seen is I've seen overgrowth of these in the GI maps and the stool tests, but also in urine testing as well. So the pH by which these other bacterial species can degrade oxalate is very, very narrow. So outside of that range, they won't break oxalate down. Now, ox factor can degrade under any physiological condition, and it's really considered a primary keystone species with the gut, and should be breaking down our dietary oxalates and allowing us to eat these foods without issues. However, that particular bacteria is tremendously vulnerable to disruption by many antibiotics, specifically things like tetracyclines and doxycyclines, but also augmentin, and zithromycin. And these are commonly prescribed antibiotics, even with children. So as a result, we do see a huge amount of people who, especially in the Lyme community, who've taken these antibiotics, they're dealing with high oxalates as a result of the treatment that they have done for Lyme. Next is mold exposure. Aspergillus and penicillium are going to be the most likely culprits, and it's not sufficient for it to be an environmental exposure. It does not have to be colonized within the system to create a high oxalate presentation. Next is glyphosate, which is unfortunately everywhere in our environments, in our landscaping and farming communities, and in our water and our food supply, and it does break down into oxalic acid. The majority of it is going to be excreted and non-metabolized, but Stephanie Seneff, whom I actually spoke with, has some really provocative research indicating that the greater the glyphosate exposure, the greater the resulting oxalic acid burden on the body. Next is poor blood sugar dysregulation is the one area a lot of people don't know, and it's very common. So many people I know that have either high or very low blood sugar, like the hypoglycemia. And there's multiple mechanisms that happen in this way, but impaired gluconeogenesis, which is the biochemical process that your body produces glucose from non-glucose sources, generally when you're fasting, like overnight. This can upregulate oxalate-promoting enzymes. And this is the process of your body making sugar from non-sugar, like I just said, when glucose levels are low to supply the brain with what it needs to do its job. Now, oxalates can impair gluconeogenesis. So that's one way that it can affect your blood sugar. But then you can also have upregulation of oxalates when there is a sensation that your body is starved of sugar. Let's say if there's a blood sugar dysregulation because of poor glycolytic pathways. And a lot of that's nutrient-based as well. And I see this rampant when I run Nutri-Eval testing. I can see how badly these patients are with their blood sugar regulation. So you can see oxalate is impairing your body's way to make more sugar. So you end up with mitochondrial dysfunction, brain fog, brain damage. So you're basically starving your brain of energy, and eventually that can lead to organ damage. And finally, there's poor digestion and bile insufficiency. I just spoke of that, but we have basically an epidemic of poor digestion everywhere, and it's often due to many factors, but when the gut lining is damaged or impaired, you're just not going to get the production of these enzymes required for digestion. And this includes things that, break down our fats, such as lipase, but also enzymes like diamine oxidase to break down histamine. And then you have elevated histamine, which damages the gut even more. And then taking this further, we see liver impairments and gallbladder dysfunction as well. And then we're unable to emulsify our fats, and it's just this vicious cycle. And we end up with these fatty soaps that bind up our minerals that are supposed to be binding up our oxalates, and we end up with this storm of passive absorption of oxalates through the GI tract. And I actually have a blog on this on my website called Enteric Hyperoxaluria. So it is the perfect storm. So there seems to be an interesting blood sugar component to oxalate that I want to expand upon, and I find it fascinating. There are two sides to this problem. So on the one hand, we have gluconeogenesis, which is, again, I said that occurs in fasting, or also it can occur not healthfully, of course. If there's blood sugar dysregulation, you can be, in gluconeogenesis because the body senses that it's starving, the brain senses that it's starving. This happens when there's blood sugar dysregulation, such as, for example, just say type 2 diabetes. But you can even have that if you have a hypoglycemic situation. But anyway, this process can upregulate oxalates enzymatically, and pyruvate, which is glucogenic in a fasted state, can also be used to make more glucose. And these glucogenic amino acids make more glucose via pyruvate. So then when that occurs, they can convert to glycine, and then from there you can get glyoxylate and then oxalate. Now, so here you have gluconeogenesis upregulating oxalate production, but then you can also have oxalate can then downregulate gluconeogenesis. So it's like this vicious storm. So that's when you get this reactive hypoglycemic state, because when you're fasting, you should be able to create glucose from non-glucose sources, and if gluconeogenesis is downregulated, then you're going to have those symptoms of brain fog and fatigue and all those other things. So this does point to blood sugar component. It's not very clear exactly how, but we do see some clues. And finally, we have also seen inhibition of the gluconeogenesis via inhibition of pyruvate carboxylase. And Chris Masterjohn talks a bit about this and the connection with biotin. So that's really, really an interesting finding as well with that particular enzyme because it is biotin dependent. Now, I did find out myself that I do have polymorphisms in biotin metabolism. And I also did a stool test, and my gut doesn't make good biotin. And lo and behold, on my NutriBel, I had a biotin deficiency and high candida, and all of that is related. But I'm getting off topic. The last point I want to mention is that glyoxylate that I just mentioned can become oxalate is a better predictor of diabetes than fasting blood sugar. And I obtained that information from Dr. Rossenberger, who did an amazing webinar on oxalate connection and just oxalates in general and how it affects your biochemistry. So I often use the phrase test, don't guess, and this is why I love medical nutrition therapy and lab testing, is you can really see what some areas that can use attention to correct. And as you can see in this image, this person is dealing with an oxalate-related disorder that can be caused by both blood sugar dysregulation and oxidative stress. So blood sugar dysregulation that I said can upregulate an enzyme known as alanine glyoxylate aminotransferase. And this can produce oxalates endogenously. And oxalates can also dysregulate a blood sugar process known as gluconeogenesis. And that's why many people that I see have symptoms of high and low blood sugar. They often can have elevated ketones in their urine and then dysregulation of carbohydrate metabolism. As you can see in this example, this person is showing various areas of needing support, such as chromium, which is needed to deliver insulin into the cell, and then magnesium, which is a nutrient that's involved in glucose metabolism. And you can also see the sulfur here, in this case, is trending on the low side. So that may be low levels in the diet, or maybe it's being used up and exchanged by oxalates, but I also see sulfur high as well for some people. And you can see this person has borderline high levels of glycolic acid, which confirms the blood sugar connection, but higher lipid peroxidation. And we can see that the oxidative stress here can also be pushing the glycolic acid. And then looking on the right over here, you can see some imbalances in carbohydrate metabolism, potential ketosis, which is the beta hydroxybutyric acid, which means this person is definitely experiencing dysregulated blood sugar metabolism because they're not on a ketogenic diet. So that's a clue there. And they're not using their sugar properly, and hence upregulation of gluconeogenesis and ketosis. And they all need to be corrected nutritionally before this person can see a decline in their oxalate production. And this makes sense, as I've seen many patients with dysregulated blood sugar, and they have the ketones. They also may have lactate dehydrogenase enzyme might be generally in this case low. Lactate might be showing up on the labs. There could be elevated pyruvate. And then the glycolytic markers are dysregulated, and they do have symptoms of reactive hypoglycemia. For example, hangry between meals, or feeling lightheaded, or they fast or skip meals, or symptoms of high blood sugar or low blood sugar. So low blood sugar would be the hangry, the high blood sugar would be fatigue after meals, and then sugar cravings. So one of the first things I have to do with my patients is help them fix their blood sugar problem. It's a fundamental issue everyone or many people in the Western world have to deal with. And often this is due to toxins and stress, inflammation, poor diet. Remember, glyoxylate can be a precursor to oxalate, and according to Dr. Rosenberg, it's a better predictor of diabetes than glucose, and it's a more sensitive marker than blood glucose. So remember, oxidative stress raises glyoxylate. Oxidative stress is one of the #1 causes of diabetes, type 2 diabetes. That is the #1 cause in the literature now. So these are some of the things that, we have to do in order to fix our blood sugar. For example, digestion, improving digestion, obviously removing inflammatory foods, and including foods with lots of antioxidants, including antioxidant supplements, cofactors and minerals that support blood sugar cycles, such as, your glycolytic, your citric acid cycle, for example, and then remove sources of oxidative stress, and that may involve also detoxification protocols. So oxalate comes from the intestinal tract, and generally it should bind with calcium, but if you eat calcium before you eat, whether it's a supplement or dairy, you can protect yourself from oxalate building up. And when that doesn't happen, or we have bad gallbladders, or lipids are not absorbed, it's floating in the gut, and then we have bile insufficiency, calcium and fat bind together, and you excrete calcium and fat, and you did not absorb, and the oxalate's going to be absorbed. It's not combined with calcium, bottom line. So we do see gallbladder issues. Maybe there's a gallbladder removed. A lot of people do remove their gallbladders. It could be just gallbladder dysfunction. It can be inflammation of the gallbladder. poor digestion, which then will allow you to absorb way more oxalate than you should. And normally oxalate and calcium should combine and you should poop it out. And when that doesn't happen, then you have this passive absorption of oxalates. And most people, they just have terrible digestion. This is what we're dealing with. It's an epidemic. And then of course there's that blood sugar component that I talk about as well. All right, let's get a little bit deeper into enteric hyperoxaluria because I think it's such a big issue here. I think it's really, really a contribution as to why we're seeing so many more people who are dealing with oxalate-related disorders. So oxalate arises from a variety of dietary and endogenous sources and is an end product of human metabolism. So we talked about this already. A small fraction of ingested oxalates, according to this journal, is absorbed under normal circumstances. Under normal circumstances, without any pathologies or any imbalances, it's about 5 to 10%, and the rest of it is derived from endogenous hepatic, which means liver production. So anything that's absorbed from the diet or endogenously is delivered to the kidney where it is freely filtered and excreted as urinary oxalate. So an increase in either GI oxalate absorption or hepatic oxalate production can increase plasma oxalates and then urinary oxalates. And this contributes to many other things like interstitial cystitis or even the risk of stone formation and other kidney outcomes. Just kidney related issues as well are big. Now, enteric hyperoxaluria can result from increased bioavailability of dietary oxalate or increased oxalate permeability. And the bioavailability of dietary oxalate is determined by the calcium content of ingested food, like I just told you, because calcium can form insoluble calcium oxalate salts, and they're eliminated in the stool. So an increase in dietary intake of free fatty acids or their malabsorption in the small intestine, this is coming directly from this journal, can result in increased fat delivery to the colon where it can bind dietary calcium and increase free oxalate that can be absorbed into the bloodstream. So bile salts and fatty acids not only determine the bioavailability of soluble oxalate in the intestine, but they can also increase the local membrane permeability in the bowel and facilitate oxalate absorption. So the net effect of fat malabsorption from any cause is increased absorption of luminal oxalates in the blood. See? So any increase in dietary intake of free fatty acids or their malabsorption in the small intestine can result in increased fat delivery to the colon, which can bind to calcium and again increase this oxalate absorption. So I know I repeated that twice, but it's really important to understand. There's also a higher risk of this with the use of antibiotics that can alter the gut microbiome, and they have found that there's evidence backing up the potential role of ox factor, even bifido and lactobacilli, and other gut microbiota in maintaining oxalate homeostasis. But more studies are needed to establish the cause and effect of this relationship. So enteric hyperoxaluria is frequently observed in conjunction with digestive diseases, as you see here, such as Crohn's disease, cystic fibrosis, chronic biliary disease, and pancreatic diseases as well, and also short bowel syndrome. And this can result in chronic diarrhea that's associated with fat malabsorption, and they may result in low urine volume, low citrate, low magnesium, and then formation of stones. So what are some of the conditions associated with overabsorption of oxalates? Well, first we have kidney stones, which is the most obvious. If you're peeing out these crystals and filtering your urine, someone might grab it, see it on a slide, and oh, here it is. Figured out why you had all this kidney pain. And many people listening to this may have experienced kidney pain at some point as well. But it can be challenging to diagnose because they're nano-sized and micron-sized, and they're not always going to show up in traditional imaging. Now, there's also cataracts and a specific kind that's associated with oxalates because the tear ducts are an excretion pathway for oxalates. And there's also associations with breast cancer and oxalates because mammary tissue does seem to be particularly vulnerable to cancer and changes as a result of exposure to oxalic acid. And then we see pulmonary fibrosis and COPD are both associated in some cases, but not all cases, with high oxalates. We see associations with painful conditions like fibromyalgia or hormonal conditions like endometriosis, uterine fibroids, PCOS that are associated with high oxalates because the crystal and the precipitants themselves can disrupt the hormone, but also because it messes up the sulfation system. For example, estrogens, androgens, thyroid hormones, DHEA, and all these things need to be sulfated. And when oxalates are high, that you can see this can cause dysfunctions in these areas. And then the thyroid, there's been a number of autopsy studies where they were performed when people had Hashimoto's and the thyroids were riddled with calcium oxalate crystals. Then there's IC, interstitial cystitis and vulvodynia, highly associated with overabsorbed oxalate, and they tend to be excretion symptoms, though they are incredibly uncomfortable for individuals who experience them. There's osteopenia and osteoporosis due to the chelation of calcium from high oxalates, and we can see that in people who have specific genetic polymorphism called SPP, which unfortunately I have that one. And we also see vertigo with disruptions to the ear canal. And then we see diverticulosis and diverticulitis being triggered by oxalates because the colon and stool they are both excretion pathways. So there's a wide range of conditions, much more than just kidney and kidney stones. In fact, only 0.5% of people with high oxalates will present with a kidney stone, and then other 99.5% will dealing with some other constellation of issues. Okay, now mold. Aspergillus in particular, to a lesser documented degree, and Penicillium and a few other species produce oxalic acid as a fermentation byproduct. And that oxalic acid combines with calcium oxalates to form these insoluble needle and glass-like precipitates that are deposited at local sites. So do we see colonization? Well, in this case, we do in this particular organic acid. So look for signs of hyperoxaluria there. They can also be in the lungs, where we see fungal wall in the lungs, and we're likely to see accompanying tissue damage as a result of the oxalates. Here's where it gets really interesting. Oxalates can trigger a specific inflammatory mechanism called the NLRP3 inflammasome. So when that inflammasome is activated, it starts the cell danger response. And when our body is not able to complete all three stages of that cell danger response, we stay stuck in these chronic inflammatory processes. So as long as oxalates are present in the body, we get this continuous triggering or activation of the NLRP3 inflammasome, and it's difficult for the body to then pull out of this inflamed state until we resolve the oxalates themselves. And I will honestly say, if someone's been in this state for a really, really long time, for several years, they actually may need some additional work to reset this inflammasome, because sometimes it does get stuck. So I've had, myself, I used FSM, and I did some water fasting to promote autophagy. And there are some other tools like DNRS and those types of things to reset the nervous system that might be required. Let's talk about sulfate. Sulfate is the fourth most common electrically charged compound in our blood, as that's how important it is. And it's very common in our body, critical. Without it, our tissues will fall apart, you'll lose muscle, joints, tendons, hair, gut lining will degenerate, and your body just needs sulfur, and it forms bonds that are very strong. It gives our protein strength, and when we talk about losing sulfur, well, we're talking about a really serious health threat. So you can see on this slide, even the synthesis of glycosaminoglycans rely on sulfur, and you understand that IC is often due to the impaired GAG layer. You also need sulfur for detoxification of your xenobiotics and steroids and things like that. And so low sulfate levels, as you can see, can cause all of these certain conditions. So how do we obtain sulfur from our diet? Well, intestinal absorption is first. So food always first. So those of you listening who have specific dietary needs, you have to hear this, because I do still stand by the fact that as humans we are omnivores. So we need fruits and vegetables in our diet. So we have intestinal absorption from our diet. We have brassica vegetables. So those are things like garlic and onions, and more so cabbage and broccoli, Brussels sprouts, and then other sulfur containing foods, even your meats as well. We do need oxidation of sulfur containing amino acids, and that's where you get the methionine, and those from animal products. So your body needs sulfur, and we talk about losing sulfur, we're talking about something really serious. It's really also important, as you can see, for the mucus layer. If we don't have enough sulfur, we have inflammatory bowel, leaky gut, dead bacteria, creating immune dysfunction. So we cannot make a good mucus layer, and mucus is required for sulfate to make it strong and sticky. So our mucus, requires sulfate to make it strong and sticky. And without sulfate, this isn't going to work. So loss of sulfate means loss of mucin integrity, and that means your gut's not going to function properly. And we also see high heavy metal toxicity because of the sulfation pathway, and I definitely have seen that on both urine and blood testing as well as hair testing. We can see these other issues that can occur as well: insulin issues, weight loss, difficulty building muscle, altered bile acid concentration, poor gallbladder function, phase 2 detox impairment, and also low glutathione. So you do need sulfur to make glutathione. So again, loss of sulfate, loss of mucin integrity. It isn't a component of intestinal mucins. There's lower levels found in GI disorders, increased risk of leaky gut, increased risk of colitis, and reduced protection from various pathogens like Campylobacter jejuni, which is an intestinal pathogen. So let's dive into the sulfur and another component here with metallothioneine. So metallothioneine is an important component of methylation, and it can bind mercury and other metals and escort them out of the body via the gut lining, the brain, and the liver. Now, what you need to take from this is its expression decreases by 70%, and that would lead to heavy metals building up. The main role of metallothioneine is regulating the maintenance of cellular zinc homeostasis. And I do often see people who are often low in zinc, but also when your alkaline phosphatase levels are low on blood work, that can also point to a zinc deficiency. So what is metallothioneine? It's basically a family of small proteins containing 61 to 68 amino acids. And again, its major role is involved in heavy metal detoxification. So there are some thoughts that deficiency in metallothioneine could explain why some people respond poorly to vaccines that contain heavy metals. So its function is, its main thing is it has about 30% cysteine in it, which then can react with a number of metals, including zinc, mercury, copper, and also cadmium. But mainly, its main important mechanism, again, is regulation of zinc. The main regulation of metallothioneine is the amount of zinc that's taken in. Now, another interesting thing about this is there's actually evidence that metallothioneine gene expression is regulated by oxidative stress, and they're also involved in antioxidation. And one of the mechanisms is due to metallothione's ability to remove hydroxyl and superoxide radicals. So take home point, without sulfur, you're going to have trouble making metallothioneine. So again, as you can see here, sulfation is really important, and it's involved in detoxification and hormone regulation and molecular recognition, cell signaling, viral entry into the cell, and xenobiotic and endogenous compound detoxification. So you can see in the picture over here, we do have the transsulfuration pathway, and you can see some of the vitamins that are involved in this. For example, B6, we've got tyrosine, which is an amino acid, and then we've got molybdenum and B2 that's also involved in this sulfite to sulfate conversion. So I know I talk about molybdenum, but riboflavin is another very common deficiency that I see on lab testing. And I also have seen it on stool testing when the bacteria in the gut is not making enough riboflavin. Often that's due to low levels of bifidobacteria. So you can see the importance of this. Let's now talk about one of the phase 2 detoxification pathways known as sulfation. Sulfation is essentially a conjugation process, and it's involved in various body processes from the biotransformation and detoxification pathways to even biosynthesis of some proteins. And it's involved in a variety of biological processes, and this includes detoxification, but also hormone regulation, molecular recognition, cell signaling, and viral entry into the cells. Sulfation of chemicals such as your xenobiotics is associated with the biotransformation of basically a hydrophobic or non-water soluble chemical or xenobiotic into one that's highly water soluble, and it's excreted into the urine. So sulfation has been shown to be an important pathway in the biotransformation of numerous xenobiotics such as drugs and endogenous compounds such as hormones, bile acids, neurotransmitters, peptides, and lipids. Now, it's also involved in the biosynthesis of your GAG, and that includes the bladder and the gut. So you can think about the GAG layer, and a lot of times we wonder why it's been degraded, or maybe it's leaky, or it's more susceptible to damage, or maybe it's the infections or the biofilm infections. It could be due to poor sulfation. And again, remember, when there's a lot of oxalates coming in, you could be depleting your sulfur, and that could be one of the mechanisms of how oxalates are destroying your bladder lining. Again, people with sulfation issues also are low in hormones, particularly DHEAS, and that's the precursor to some of your other sex hormones. And we do see a lot of low hormones amongst people with IC, and it is also involved in stabilizing and storing other biosignaling molecules. Really important. So again, sulfation is used to conjugate toxins with sulfur-containing compounds. And this process is actually catalyzed by a series of enzymes known as the sulfotransferases, or SULT, salts is what I will abbreviate it as. And there's also genetic polymorphisms in this. I do see patients who have that as well. So understand if you have a polymorphism, you may be downregulated in the functioning of this enzyme. So the sulfation system is important for detoxifying chemicals, industrial, environmental, drugs, steroids, food additives, and toxins, and also from intestinal bacteria. It is used to detoxify some normal chemicals as well, and it is the main process of eliminating catecholamine, neurotransmitters, steroids, thyroid hormones, and glucocorticoid hormones, as well as conjugate bile acids. And we do often see there could be also toxic bile acids also accumulating, could be part of the problem too. So sulfation is the primary route for elimination of neurotransmitters, and we also have seen that dysfunction in this system could also contribute to the development of some neurotransmitter system or nervous system disorders. And finally, it is one of the major detoxifying enzymes for phenolic xenobiotics. All right, so I know this is going to get a little bit more sciencey, but what I want you to take out of this slide is that you need ATP to sulfate. And everyone listening has to understand, and I'm a big, proponent of ATP and making sure we're making enough, not too little, not too much, just the right amount. So the sulfation reaction needs sulfotransferase enzyme, the salt I just talked about, as a catalyst, and a co-substrate such as a sulfuryl donor. So now the universal donor for these reactions is known as active sulfate, but we're going to call that PAPS. Okay, so the activated form of sulfate is known as PAPS. And sulfation has an entire family called sulfotransferases that are available as enzyme catalysts, and they use activated sulfate, also PAPS, as a high energy donor, and it's basically the universal donor for all sulfation pathways. So the body needs to rapidly make PAPS when sulfate conjugation is required. So here's the thing. PAPS is made from sulfate, but making PAPS from sulfate is dependent on the availability of ATP, you guessed it. And we also do need magnesium, which I see widespread deficiencies of that. So if your energy processes are impaired, and we can identify that on like a NutriVal or a hair tissue mineral analysis, you will have problems with this process. So again, sulfate from PAPS is a need as a substrate for sulfate conjugation. So PAPS is supplying the sulfate. And low level of PAPS exists in the body, but are synthesized quickly as needed from inorganic sulfate. Or also, I should mention catabolism of cysteine and methionine, which are two very important amino acids. So here are some of your sulfotransferase enzymes here. I highlighted SALT1A because I do often see polymorphisms in that one. And it's found in the liver, intestine, brain, and platelets. And it really has a strong affinity for various different substrates. SALT1A1 is used for xenobiotics and thyroid hormone metabolism, and also procarcinogens as well. But it is subjected to a very common functional polymorphism. So I can see that on the tree of life. So I don't want to spend too much time on this slide because you can obviously see that sulfation biotransforms two main classes of compounds, phenols and amines. And so you can see, for example, phenols can be synthesized by a variety of chemicals, but they also occur in nature, and they're easily absorbed by inhaling, skin contact, and ingestion. So you can see some of these sources of human-made phenols are like bisphenol A and B, herbicides, pesticides, fungicides, triclosan, xylenol, and tyrosine, and they're also found in some foods. So you can just kind of go through here and see different things that they're involved or they're found in, and then the amines are also found in the body, neurotransmitters, melatonin, amino acids. They're also found in aged food, and there's also exogenous amines as well that are found in different things. For example, they're used actually to make Tylenol, which is acetaminophen, and blue dye. And there's also some that are found in antifreeze as well. and emulsifiers in many household products, even ice cream. So I had to emphasize this again because it's really important so you understand what sulfur deficiency could be doing to your bladder or even your gut, actually. So it's used to make necessary substances, and that includes things like sulfonated glycosaminoglycans, which are your GAG, but even things like chondroitin sulfate, heparin sulfate, keratin sulfate, heparin. So these are found in things like ligaments, cartilage, tendons, vasculature, cardiac valves, skin, and then all cell surfaces have heparin sulfate and the basement membrane of your epithelial tissues. So sulfate deficiency can really impact your integrity of various linings of your body and organs. And then keratin sulfate heparin is part of mast cells, and it's found in the skin, lungs, and liver. But also sulfation is involved in thyroid hormone synthesis, T3 and T4 as well. Sulfur-containing molecules are also found in the body. B vitamins, biotin, B5 and thiamine, methionine, SAMe, homocysteine, cysteine. I talked about metallothionein, taurine, glutathione, and also alpha-lipoic acid. So these are sulfur-containing molecules. So if you're taking supplemental forms of these as well, you are providing your body with sulfur as well. Sulfation and sulfate oxidase activity are different. So various sulfites are widely used, for example, in potato salad as a preservative, and salad bars to keep vegetables looking fresh. Dried fruits have them to keep, for example, dried apricots orange, and some drugs. So normally the enzyme sulfite oxidase metabolizes sulfites to the safer and needed sulfate, which is a process called sulfooxidation. And those with poorly functioning sulfooxidation systems, however, have an increased ratio of sulfite to sulfates in their urine. So to convert sulfite to sulfate in this process using the SUOX gene, the cofactor is actually molybdenum, and it's needed for enzymatic action. Now here's something you should know. Glyphosate is a known chelator of molybdenum and can be a cause for sulfite oxidase dysfunction. Other ones include B12, B2, manganese, boron, strontium are also needed to make sulfate. So people with issues in the sulfooxidation process, they're more likely to get headaches, migraines, asthma, chest tightness, chronic fatigue, and chronic indigestion and heartburn. And they may react to high sulfur foods as well, such as cruciferous veggies or mustard family, or high sulfur plants such as garlic and even eggs. The sulfate in Epsom salts could also cause a reaction if the makeup of the colonic bacteria includes more of them that change sulfate back to sulfur, and that includes people who have overgrowth of E. coli. In this case, the sulfate is changed to hydrogen sulfide and then into sulfur amino acids, and this can also lead to an increase in sulfite to sulfate ratio and low sulfate oxidase. Keep in mind if you have high oxalate levels in the body, this can also create a reaction with Epsom salts or other sources of sulfur, because they will increase sulfate in the body, and then sulfate is used to help remove oxalates from the body. Now what happens is this can cause a sudden movement or mobilization of oxalates, and that can be really painful. This is known as oxalate dumping. This is why we have to be very careful on how much Epsom salt we use when we're trying to detox or increase our sources of sulfur. And although Epsom salt is also helpful for people who are dumping, it can also cause dumping. So the amount you use is very important. Again, this would only be noticed as someone who has low sulfate and suddenly increases it. So how does sulfate inhibition actually happen? Well, there's many factors that can influence the activity of sulfate conjugation. So one is your diet. A diet low in methionine and cysteine, two amino acids, are used as building blocks to make sulfate, and low levels of that can reduce sulfation. But there's other things that are going on as well. There's a process or a situation known as competitive inhibition, and this is when there's more than one compound that's transformed by a single enzyme. So if that happens, if in this case one compound is unable to be transformed due to the competition of the other. So let's do an example here. If a person is around a lot of essential oils that have phenols and they're eating a lot of high phenol foods, they may end up without enough sulfates for sulfation. So they're going to run out of the enzymes. So if the person has a high oxalate problem, they're going to run out of sulfates, especially if they have hyperoxaluria, whether it's outside or inside sources for that, and it's not being treated, they'll definitely be reacting to foods with oxalates, but also phenols in them as well, and any essential oils or chemicals that contain phenols that are floating through the air. And this is oftentimes when you see people have chemical sensitivities. An increase in toxins from the environment or even our bacteria in our gut can also lead to what's called inhibition, and this is just due to overburden of toxic load. Well, maybe it's not inhibition in the sense that that's what researchers call it, but it's really just the availability of enzymes because you're using up your available enzymes faster than they can be made. So, for example, bacteria and fungus such as candida and yeast can also decrease availability of sulfation enzymes. Now, another mechanism for this inhibition or depletion is your depletion of your necessary cofactors. So sulfation is really susceptible to inhibition due to depletion of cofactors, and there's a delicate balance of your serum sulfate concentration, and this does rely on absorption of inorganic sulfate, its production from cysteine, and the elimination in your urine. So over a 24-hour period, sulfate levels can really dramatically change in a person. They can decrease if you're fasting or eating a lot of substances that are metabolized by sulfation, such as, you guessed it, Tylenol or acetaminophen. A person excretes about 20 to 25 millimoles of sulfate in a 24-hour period. So they have to constantly be maintained by your intake of sulfur-containing amino acids, but also inorganic sulfates. So when sulfation is inhibited, a person is unable to detoxify. So, for example, it'll be difficult to eliminate Tylenol, but also some neurotransmitters from the brain. Two things that can inhibit sulfation. One is, we said acetaminophen, which is Tylenol, but also aspirin can, like non-steroidals, and also yellow food dye. So how does this affect oxalates? Well, I'll explain. Sulfate and oxalate are bidirectionally exchanged across the membrane. So for every sulfate ion crossing one way, an oxalate passes in the opposite direction. So high amounts of oxalate on one side of the membrane can pull large amounts of sulfate out from the other side. And so in the context of kidney filtration, and high body oxalate burden can cause us to dump sulfate in the urine while retaining oxalates. And I definitely have seen this on a test known as the metabolomics. So I spoke a little bit about how sulfate and oxalate are bidirectionally exchanged previously, maybe briefly, but this is really important to understand. I think it's an important piece of the oxalate story, and a lot of these knowledgeable practitioners, they just miss this piece that's so important about the fact that oxalate precipitate itself, because that's what they think mainly about, but they're missing the whole association with how it affects different biochemical processes in the body. And this shift with the sulfate and oxalate is very, very profound. So again, for every sulfate ion crossing one way, an oxalate ion is going in the opposite direction, and oxalate also outcompetes sulfates. So it's almost like it's stronger. So as long as it's present in high levels of the body, it's very difficult to maintain adequate sulfate status. So the vegans, listen up. If you are eating a very high oxalate diet, loaded with high oxalate foods, or if you've got a mold burden in your GI tract, you might be finding out that you're impairing sulfation, and that can affect so many other things. For example, I mentioned hormones. It could also impact cholesterol, which needs to be sulfated. Again, I said DHEA, thyroid hormones, which need to be sulfated, and I do see sluggish thyroid on many tests, thyroid testing, as well as blood serum and hair. Reproductive hormones, cortisol needs to be sulfated, heparin, so these components of your bladder lining need to be sulfated. We can measure urine sulfate levels to assess whether this is happening, or you can do a dipstick for sulfate levels. There are some companies out there that do this. And these are mechanisms by which this is happening. And then we have also the SLC26A10, which is a whole other thing. It's a solute carrier for sulfate, and it's involved in the exchange of sulfur and oxalates. So So basically, oxalate competitively inhibits the uptake of sulfate in cells. It outcompetes sulfate. So there's also a few tremendously toxic mycotoxins that need to be sulfonated to be cleared from the body. So these two that are listed here are two of the top ones. T2 toxin is from trichothecene, and so they're starting to be regarded as two of the most toxic mycotoxins that we can be exposed to. So anytime these are present, even though they're not being produced by organisms that produce oxalate, if we have high oxalates because of, let's say, fat malabsorption, like I said earlier, or high oxalate diet, it can create a significant roadblock to the clearance of these two particular super toxic mycotoxins. So on the one hand, you can get oxalates from the presence of mold, but then you can also have inhibition of clearing mycotoxins by the presence of oxalates. So it's kind of like a vicious cycle. Now, again, like I said, everything we think about that needs to be sulfated, such as benzenes or acetaminophen, certain mycotoxins as well, catecholamines, androgens, estrogens, thyroid hormones, bile acids. There's a tremendous list of both endogenous and exogenous compounds. And now you can see what it can happen when oxalates are high. When we have a problem with oxalate, we have a problem with sulfate, period. They use the same transporter. So I've picked up this slide from this YouTube video, The Downside to High Oxalates. Very, very, very interesting and eye-opening. So when oxalates are present in the urine, they'll get reabsorbed from the urine while we lose a molecule of sulfate. So in the kidney, they're absorbed in exchange for sulfate. So the more oxalate in the urine, the more stones you have and the more sulfate you will lose. But not just in the kidney, this happens all over the body. So whenever there's excess oxalates on one side, they will activate the receptor and cause sulfate to be removed. This is just the chemistry of our bodies. So in the liver, stomach, large intestine, we have these receptors, and sulfate moves one way, oxalate moves the other way, and they're exchanged for each other. So if the blood is loaded with oxalates, oxalates will go in the liver and sulfate will go out, starving the liver of sulfate. So bottom line, if you absorb a lot of oxalate, you lose a lot of sulfate. And this can also occur in the colon and the small intestine, same thing. So if you have good sulfate levels in your diet, you absorb sulfate and get rid of oxalates out of the body. If oxalate in our gut is floating around in high concentration due to poor digestion, oxalate leaks in and sulfate gets kicked out. When we have a diet high in sulfate, we absorb sulfate and get rid of oxalates. So oxalate and sulfate go in different directions, and this is because of these transporters. Blood levels of sulfate are controlled by the kidneys. So if we lose sulfate in our kidneys, this is where the problem begins. Oxalates can antagonize the kidney, and is the kidney is where we lose our sulfate. So when oxalate builds up in the blood, oxalate and sulfate go in different directions. So if you end up with a lot of oxalates, again, it'll pull the sulfate out of the body in the urine. And this is important for anyone who wants to overcome their chronic health challenges. Here's a really good image here that describes this as well. The arrows are reversible, so they're not stuck in that one direction. So you have this SAT1. It's a transporter protein and can get dysfunctional when oxalate builds up. And then we have the NAS1, which is a sulfate transporter that to get it from the urine into the blood. So it takes the sulfate in. So again, with high oxalates in the urine, sulfur is lost in the urine in high amounts. So, and there is a study that actually found that bacteria without sulfate transporters crush the sulfate transportation. And understand that low sulfur will affect everything. So low sulfur in the blood often is correlated with high sulfur in the urine. So low sulfate contributes to 12% reduction in 5-HT in the brain, and this can actually lead to depression. So serotonin comes from 5-HT through this tryptophan pathway. B6 is a cofactor as well, and also keep in mind that B6 often is seen as depleted when there's a lot of inflammation in the body. But there's other reasons why B6 is depleted. It's very sensitive to certain antibiotics, alcohol, as well as even a lot of SSRIs and medications, and birth control as well. So norepinephrine, another neurotransmitter, and serotonin are also sulfated. And so when sulfate is low, the brain does become deficient in neurotransmitters. Now, blood levels of serotonin can go up as well, and this can lead to serotonin syndrome. So again, oxalate problems create sulfate problems, and they can create neurotransmitter problems. So a lot of times when people come to me and they're suffering with depression or anxiety and they believe it's due to their pain, I actually will argue that it could actually be due to this as well. So let's talk about hormones in a low sulfate environment. So hormones take a lot of energy to produce, and they're chemical reactions to make cholesterol, cortisol, aldosterone, pregnenolone. And when the body likes to store hormones when it is needed, sulfur is a major requirement for that. So unfortunately, when sulfate is low, it does affect hormone balance, and I can definitely see that. You can see that here in this kind of situation. So we've got low DHEAS, which is a sulfated form, and then you can see because DHEAS is needed to make testosterone, you can see that this person has low testosterone. People who have oxalate problems, the body loses the ability to store hormones by sulfating it, and then the hormone is lost in the urine, and that lowers reserves and makes us have higher requirements for certain nutrients to help replace the hormones we're losing. So hormones have a very important relationship with sulfate. If you cannot sulfate them, you can't hold on to them. So with people with oxalate issues, they really need to add sulfur or sulfate in the forms of supplements or baths slowly. And this will help remove the oxalates and can bring on a painful crisis as the oxalates are moved around. And these people need to start really slowly. So with this issue is also seen when using amino acids or supplements such as N-acetylcysteine, NAC, which is very commonly recommended. They do supply sulfur compounds, and the problem is the body needs to change the sulfur first into sulfite through that sulfoxidation reaction, that sulfo-oxidation reaction, and then using sulfite oxidase, SUOX, it has to change the sulfite back into sulfate. And this step does involve various cofactors, particularly molybdenum, and I mentioned earlier B2, but I didn't mention that also B12, manganese, boron, and strontium are also used. So I do see a lot of boron deficiency on hair testing, and I do see a lot of B2 on my blood work or some of the Nutraval, and then I do see needs for manganese as well. And sometimes manganese can also become biounavailable in certain circumstances when there's excretion issues or slow oxidation rates. Another way to supply sulfate is through Epsom salt baths, which do contain magnesium sulfate. And I do like that because magnesium is also really supportive in the body. So here are some supplements that contain sulfur. We've got the alpha-lipoic acid, chondroitin sulfate, glucosamine sulfate, glutathione, indole 3-carbinol, which actually can convert to DIM, which is often used when there's estrogen dominance or high estrogen, MSM, methionine, NAC, and taurine. Now what's interesting is I've had reports of people saying when they do a really high sulfur situation, they don't tolerate it well, and this is known as sulfur intolerance, and that often has to do with dysbiosis, but that could also be due to lack of cofactors for some of these processes. Now I also have experience where someone will start something like DIM, and they may start having symptoms similar to menopause as well, which is hot flashes, and it's not actually causing menopause, it's probably causing sulfur intolerance, which one of the symptoms of sulfur intolerance is hot flashes. So how do we induce sulfation? Well, anything that will enhance the production of cysteine, which is the building block to make sulfate, is going to induce sulfation, as you can see down here. Now, NAC is a supplement that can be used as a substrate to make sulfate, and it does involve multiple steps needing multiple enzymes, and that does actually include iron, which, by the way, if you have copper toxicity or copper dysregulation, you could have an iron deficiency, and I see this a lot as well. Tyrosine, which is an amino acid, B6, which I see tons of deficiencies of, and B2, and also molybdenum. Very common deficiencies within my patient population. So here's a diagram below that shows the oxidative and non-oxidative pathways for cysteine to become sulfate. Take-home point is be careful about taking any product such as NAC that is a chelator, as they can chelate out toxins such as mercury or mycotoxins. If you take too high of a dose or do it sporadically, it can cause symptoms from pulling out toxins in an unconscious and really haphazard manner. I think a lot of people are taking way too much NAC. A little bit is okay if, like, you know, it's in a supplement, like 500 milligrams, but I think people taking 2-3 grams of it, it's a little risky. Now, not shown on this chart is the production of hydrogen sulfide. That's toxic in excess, but necessary in small amounts. And this occurs when homocysteine is changed into cystathionine, as you can see here, using B6 as a cofactor. And again, when cystathionine is changed down into cysteine, again, using B6 as a cofactor. This molecule, hydrogen sulfide, travels through the body tissue by simple diffusion and moves around, and it's involved in blood vessel relaxation and neuromodulation and regulation of inflammation and insulin and energy metabolism and cardioprotection. But anyway, it contains inorganic sulfur and can be oxidized as a source of sulfate. So the process of clearing excess hydrogen sulfide is protective to the body while supplying the much needed sulfate. The mitochondrial sulfide oxidation pathway is coupled sulfide catabolism to oxidative phosphorylation, and this makes sulfide an inorganic substrate for human electron transport chain. So this is yet another way where your sulfation pathways can actually help you produce, you got it, ATP. So let's shift back to the idea of colonized mold creating high oxalates. I mentioned earlier that we want to think about looking for the damage where we see that colonization. And in this study, you can see here, there's been numerous calcium oxalate crystals were found in the cavity wall. And the oxalic acid produced by the Aspergillus, in this case, Aspergillus niger, was the main cause of the patient's fatal respiratory failure. And this is, you can see a lot more than just kidney stones. And it's not trying to minimize the effect of kidney stones, but oxalates can potentially be fatal. People are just not aware of it. And so there's been other case reports of invasive Aspergillus niger with fatal pulmonary oxalosis. And in this case, there was a fungal ball, and the fatality was a result of the oxalic acid being produced by the colonized mold. Now, I'll say that we're going to find that oxalates when we find colonization. It's mostly this is what is occurring, but it's not exclusive, just an FYI. Here's another case looking at renal oxalosis and a fatal version of it. So the oxalates were being produced by the colonized mold in the lungs, and those calcium oxalate crystals mobilized and affected the kidneys. And so we can see how oxalates in certain places other than the colonization can actually travel, but the colonization is where we should be looking first, and that's usually a GI tract. So let's get into the genetics of oxalates. Like I said, this is going to be the minority of patients, but in my practice, I do see it about 15%, which is very significant for these people. So the four primary ones are GRHPR, SPP1, HOGA1, and AGXT. Okay, the first one is GRHPR, and this is the glyoxylate hydroxypyruvate reductase. And we're going to call it GRHPR. And this enzyme converts glyoxylate to glycolate, and glycolate is easily eliminated from the body easily. But when we have polymorphisms of the SNP, the conversion does not occur properly, and glyoxylate builds up and gets converted or broken down into oxalic acid, and then we form these oxalic acid precipitates. So glyoxylate should get converted to glycolate, and glycolate can easily be used or excreted. But when that doesn't happen, we get the shift to oxalic acid. And I should also mention that NADP is used as a coenzyme when we're making oxalate. So NADP is a really big factor in mitochondrial energy production. And so when it does get depleted, and that happens a lot when there's mold overgrowth or toxicity in the body, or even age can cause NADP or NADPH to be depleted, it's a really big factor in the mitochondrial energy production. And so if it does get depleted, especially with this, we have this excessive oxalic acid production, it becomes a secondary mechanism by which we see oxalates are associated with chronic fatigue syndrome. Now, if you see a homozygous polymorphism, which would be a 2 here, these are heterozygous, so there's only 1. If you see 2, in this case, I'm looking at the tree of life here, right? So that's what they tested here. When we see homozygous, we have a much more strong association with this endogenous oxalate piece. Also, I should mention that those people who have the two with some of these, especially this and the AGXT presentation, it's definitely much more significant on the influence of the corresponding enzyme. If you have only one, I think it's still important to be aware of it, but I don't think it's as severe. Here's the next one. This is called the AGXT, called alanine glyoxylate aminotransferase, and it's mainly found in liver cells, specifically within peroxisomes, which are important for removing toxic substances from the cells and helping break down certain fats. This enzyme converts glyoxylate to glycine, and variations in this gene can lead to type 1 primary hyperoxaluria. And again, it's more profound when there's a 2 there. And this is our primary kidney stone formers, actually. So then some genetic analysis programs, you'll find this coded as just AGT, but it can be seen as both AGXT or AGT. So again, this is alanine glyoxylate aminotransferase, and it's going to lead up to the buildup of glyoxylate when there's a homozygous polymorphism primarily. So when this enzyme's compromised, it's when we'll see our type 1 primary hyperoxaluria. I should also mention this enzyme is B6 dependent as well. So if there's B6 deficiency, it can reduce the function of this enzyme. So if you have a polymorphism and you have B6 deficiency, it's a double whammy. Again, these are our primary genetic kidney stone formers, but about 0.5% of people with high oxalates will actually develop kidney stones. So I don't know how relevant that particular association is. Again, this is homozygous presentation for that severity. It's fairly rare even to see a heterozygous polymorphism with excessive kidney stones, but I do believe, and I have seen with people with like other oxalate-related conditions like interstitial cystitis, if they have a 1 there, I have definitely seen that there was a strong association with that condition. So maybe not so much the kidney stones, but I would say with other certain conditions related to oxalates, the 1 would be impactful. So here's the one I have. This is the SPP1, which is involved in the attachment of osteoclasts in these cells in your bones to the mineralized bone matrix. And this encoded protein is secreted and binds hydroxyapatite with high affinity. So there's another protein called osteopontin, which is a critical inhibitor of calcium oxalate crystal formation and retention. This protein is also a cytokine. It can also upregulate the expression of interferon gamma and interleukin 12. So studies have shown that the GG genotype for RS2853774, which is this first one here, increases the likelihood for calcium oxalate urolithiasis. Now, again, I said it's involved in the attachment of osteoclasts to the mineralized bone matrix. But unfortunately, it's also seen with things like osteoporosis and osteopenia, which my mother has, which, you know, it does concern me about my predisposition for that as I get older. But also, again, it increases the likelihood for calcium oxalate urolithiasis, which is probably why I had all the urinary pain and also a lot of UTIs in the past. This is an area where I think mineral balancing can be really helpful. As I forgot to mention with the SPP1, because there's an increase for the calcium oxalate urolithiasis and attachment of calcium issues, individuals who have the SNP should have plenty of vitamin K2 to help with the attachment of calcium. But I also think just overall balancing the minerals, as I see with the hair testing. So here's an example of my hair tissue mineral analysis. In the beginning, my calcium was at 149. So it wasn't fully a shell, but it was definitely biounavailable calcium. And I did some mineral balancing in K2, and I was able to get it down to 130, as you can see, because of my genetics, it's still pretty high, but it's coming down. So my goal is to try to get it into the purple line over time. And so, and one of the ways to do that is to support these first 4 minerals, the calcium, magnesium, sodium, and potassium. So this is another reason why I run the HTMA with all of my patients. And finally, the HOG-A1 gene encodes the mitochondrial 4-hydroxyl-2-oxyglutarate aldolase, which catalyzes the final step in the metabolic pathway of hydroxyproline that releases glyoxylate and pyruvate. This gene is expressed in the liver and kidneys, and variations may lead to hyperoxaluria type 3. Not very common. Actually, I have not yet seen one person with this one. I've seen the other 3, but not this one. So this is one I'm showing you, which is HOG-A1, and this is going to be involved in our hydroxyproline metabolism, which again, I said, it allows for the release of glyoxylate and pyruvate, and this is our newest association with primary hyperoxaluria type 3. And individuals with this presentation can have issues when they either have a heterozygous in this case or homozygous. You have a 1 or 2, which often gets overlooked. And because of this hydroxyproline and pyruvate relationship, individuals with this one do very well with inclusion of high-dose thiamine. Again, another reason why I really love thiamine to be used therapeutically. But again, the heterozygous and homozygous presentations are significant. So they don't just come from food, oxalates, they're made within our cells or biochemical byproduct of our metabolism. I think this is another area that's often overlooked by, I think, a lot of the people in the oxalate community, but even in the carnivore community. I think they're so hyper-focused on the diet and they're forgetting the biochemistry that's going on behind the scenes. So when we're healthy, we're balanced and they're metabolized correctly, and most of us are living far from optimal lives. So looking at oxalate is an extremely important step. And again, you don't have to have bladder pain syndrome for this to become a problem for you. It can affect anybody. So glycine, which is an amino acid found in a lot of protein and supplementation, can get converted to glyoxylate by another enzyme called DAO, and this is not your diamine oxidase enzyme, by the way. It's a different enzyme, and this can actually convert to oxalate as well. So this is our glycine oxalate pathway. It's occurring inside of your liver cells. You can see here the process that occurs. You have gluconeogenesis, which I talked about with the blood sugar, and then this, then you have serine that's going through this AGT enzyme, and then alanine again, and then we have glycine. Then via DAO, you have glyoxylate, which can then convert to oxalate. Now, this particular polymorphism or gene is often seen in conditions such as schizophrenia, bipolar, there's the primary hyperoxaluria, autism, and also Crohn's disease. So how do we determine primary hyperoxaluria or secondary? Well, the NutriVal and also the Great Plains Lab right now has these 3 markers, and I believe the NutriPath also does now in Australia. And it's a really good tool to kind of take a peek, but keep in mind these are urinary excretions, and you have to be excreting oxalates at the time that you collect the urine. Remember, oxalates are stored in the tissue. So if you're not excreting it at that time, it does not mean you don't have an oxalate problem. I have to repeat, it does not mean you don't have an oxalate problem. So this is a screenshot from the NutriVal, which is from Genova. And you can see this presentation here. What is it saying? Well, in this presentation, we might have a primary hyperoxaluria expressing with the elevated glyceric. But then we have elevated oxalic, which could also indicate that there is also secondary hyperoxaluria coming from diet or from mold exposure. So in this particular case, this person could be having a combination of genetic or biochemical oxalate accumulation or excess, and also exogenous. So it's not always just from genetics. It could be one or more factors. It could be this person has also leaky gut. It could be fat malabsorption. It could be overexposure to glyphosate. It could be colonized mold. It could be all of the above. So I'm just highlighting this one more time. The 2 primary pathways, the GRHPR and the AGXT, when either or both of these are not functioning, we do see high oxalates produced. And so when we see glyceric elevated, like in this case, we want to look for that GRHPR, and when we see glycolic, generally want to look for the AGXT. And we can get a little clue because this also inhibits enzyme called tyrosinase, which can then suppress melanin formation, and sometimes people will have lightening of the skin, vitiligo, or other clues that can be giving you signs of type 1 hyperoxaluria. As I mentioned, when oxalic acid is the only one elevated, then it's probably not genetic, it's only from outside sources, most likely. And I mentioned many times before. So that's why I really love this test, because it gives me a lot of insights, not only about oxalates, but also your overall biochemistry. Also, another thing people don't realize is high dose vitamin C, which is more than 250 milligrams at a time, can also contribute to oxalate burden. Some of the organisms in our microbiome can convert vitamin C into oxalic acid. So you'll see people say, oh no, vitamin C doesn't convert to oxalic acid, but in human cells, there are in human cells, that's not what's happening. So yes, your cells can't convert it to oxalic acid, but your microbiome can. They convert it to sorbic acid and then oxalic acid. So if you're taking more than 250 milligrams at a time, you could run into high oxalate production. And I know after COVID, a lot of people were like downing, grams and grams and grams of like 2, 3, 4 grams of vitamin C, and that can really be problematic. So, and finally, you do want to think about mold, and then very rarely, but antifreeze poisoning can result in elevated oxalic acid, and also using Miralax, which is polyethylene glycol. So a lot of different things that can contribute to elevated oxalic acid. But again, genetically, you would more likely see glyceric and glycolic. So Aspergillus species in the GI tract can produce exogenous oxalates. I said that many times. So individuals with elevated Aspergillus frequently have a subsequent elevation in oxalic acid, and elimination of fungus and mold is important for reducing excessive production. So what do you have to do about it? Well, you have to fix the gut. So many of these mechanisms by which we overabsorb oxalates are gut driven, and if we have colonized mold, yeast, or fungus driving it, we got to get rid of that if you want to ultimately get rid of oxalates. So if we're dealing with overabsorbed oxalates because of leaky gut, we need to work on healing and sealing the gut to avoid that progression through the intestinal lumen. If we're dealing with poor fat emulsification, we need to provide biosupport and digestive support. And in that part, but not all of our interventions should be dietary. And this is exactly why going to your medical doctor, whether it's a urologist or a GI doctor or whoever, a primary care physician, is probably not the person to go to when you're trying to resolve this condition, because it doesn't involve medications and surgery. It involves diet and lifestyle. So this is why nutrition and nutritionists and functional nutritionists like myself are really, your first line of defense in trying to heal from a condition like this. So here's your low oxalate diet. You should have to go slow and very slow to avoid that mobilization or dumping of oxalates. So generally the recommendation is about 5 to 10% weekly, and the goal for chronic stone formers is no more than 50 milligrams of oxalates a day. So I should repeat, if you're a stone former, 50 milligrams a day is really the upper limit, but it's really rare for most people to have to be this strict. So really, everybody else, avoid the big four: spinach, Swiss chard, rhubarb, beets. And then, I kind of look at it like you can have a little bit of oxalates here and there. Just try to center most of your meals around low oxalates. If you're eating plant-based foods, low oxalate foods, and obviously you want to have adequate protein in the diet. So I'm not going to tell you to follow a carnivore diet, obviously, but I'm more of a believer of omnivore. But of course, you're going to want to go and make sure that you're not loading up on high oxalates foods. So there are people in the high oxalate community that think it should be a long-term intervention. I have to tell you personally that I am actually handling oxalates a lot better now since I started working on my gut. And I don't eat a lot of oxalates in my diet yet, but I'm definitely not completely avoiding the foods. So again, I truly stand by the fact that if we can identify the underlying mechanisms and take the appropriate steps to correct them, we should be able to normalize the diet to some extent. And so while we're trying to do that, we do need to temporarily reduce the amount of dietary oxalates. And for some people, it will be lifelong. And we want to make sure we do this slowly so that we don't get into what's called oxalate dumping. So what are your oxalate dumping symptoms? Well, they're listed here. And there's just a bunch of them. I mean, it could be any of these or other things. So if these kind of symptoms are getting worse or new ones are popping up when you're trying to embark on a lower oxalate lifestyle, then you should blame dumping, and also known as mobilization. And it's not something that's in the science literature in detail, but it's very anecdotal, but so many people have experienced it, and this includes myself. When I went through dumping, it went on for about 6 months, and I was having more UTIs, and I was actually having blood because of the crystals that were coming out and irritating my urethra. Here's just a list of some more symptoms that can occur. Obviously, they're not all bladder symptoms. You can see there's so many other things that can happen. Tinnitus is a very common one. Headaches, joint pain, peripheral neuropathy, big one. Back pain, cold sores, things like that. Okay, so here's another one. Now, one thing you can do is to use calcium to your advantage. So when we want to use that affinity of oxalic acid to calcium to our advantage, we don't have to be as strict with the diet per se. But again, It's all individual. I still think it's smart to stay on a lower oxalate diet for the most part, but I think there's high variability amongst each person depending on what your circumstances are. But calcium definitely helps. And calcium citrate is very helpful. So if you have someone who can't tolerate citrate, well, there's other forms of calcium that will work as well. Sometimes I'll just drink a big glass of flax milk that has about 400 milligrams of calcium and mix that and make that drink while I'm eating something. Calcium can be used as a binder for oxalic acid, and we could put that extra calcium in the gut at the same time. Then we can put the oxalates. When we put the oxalates in the gut, it should bind. But again, don't do too much because obviously there's a limit to the amount that will bind. So I just think it's all about balance. So the goal is to take the calcium more than 30 minutes before you eat, and if you're not getting so again, anywhere from 30 minutes or so prior to eating with the meal will help the calcium bind the oxalic acid and let it be excreted harmlessly from the stool. Magnesium can also be used as a binder in much the same way as we would use calcium. So magnesium is almost as effective as calcium. So you can do like a CalMag as well. And this can reduce oxalate absorption and increase urinary excretion. So if we bind it in the gut, it allows the oxalates to be filtered through the colon and leave the body through our stool rather than getting in the bloodstream and being filtered by the kidneys and leaving in that way. So a little bit of calcium as a binder, a little bit of magnesium, and watching the oxalates on your plate are really, really smart strategies to help remediate this situation. Don't go crazy trying to do this. I don't think it's necessary to count all the grams of oxalates because I just don't think that's effective. So, and again, there has to be a good oxalate and calcium ratio as well. You don't want to take too much calcium because then you might absorb it. So I usually say about 400 to maybe 500 milligrams. Now you do need about 1,000 to 1,200 milligrams a day. It's just for general health. And again, magnesium is also important too. I should also mention when you're using the magnesium, it's not for the bioavailability of what we're trying to do for energy or for blood sugar. That's magnesium glycinate is a better source for that, or magnesium malate. But in the case of binding oxalates, the citrates, even though it's the less bioavailable, it's the best one to be using for binding up the oxalates. So calcium citrate, magnesium citrate, and even potassium citrate can help protect the cells from oxalates and calcium oxalate crystal induced injury, but also decrease lipid peroxidation and reactive oxygen species. And another one I have to mention is B6. It's another very important nutrient for reducing oxalates. Again, I told you the AGXT is the mechanism, and it's B6 dependent. And I talked about this earlier. So remember, glyoxylate becomes glycolate. Glyoxylate becomes glycolate, and this uses B6 as a cofactor. So if you have genetic polymorphisms, B6 can be tremendously helpful. But again, be very careful because it can also induce dumping. And I have also experienced people who have other enzyme issues like the HDC through histamine. It can actually provoke more of a histamine reaction. So again, everything should be done carefully. But B6 is so important. It is also used in transsulfuration. Remember, we talked about endpoint as being sulfate. So again, B6 can help support the sulfate, which can then help support the oxalates. So what are some of the interventions that you should be doing? Well, first, you should test and don't guess, because if you guess, you're probably going to feel like a hamster running in a wheel and getting nowhere. So it's really important to understand what's causing the dysregulation with oxalates. So I start with a series of tests, and of course there's others, but these are the ones that I have found to be the most beneficial. And of course it also depends on your location, where you're located, on the availability of these tests. But here's a snapshot of some of the tests that I do. So I talked about mycotoxins being a source of oxalates, especially aspergillus. So obviously a mycotoxin profile is important. And then there's the glyphosate, because remember I mentioned that glyphosate can also convert to oxalates as well, amongst other immune disruptions. The hair tissue mineral analysis, checking mineral status. Is there low minerals? Is it biounavailability? How is it affecting your adrenals? A lot of times oxalates can cause a depletion of sodium and potassium, which then you can see affects the first two minerals, calcium, magnesium, and leads to biounavailability. But then there's also stress response from inflammation can also raise sodium as well. And so it's important to be able to balance that out. And then the iodine testing is super helpful because that one is used just to check iodine status as it supports the thyroid, and the thyroid is very important for your immune system and your metabolism as well. But in addition to the iodine, I also check for halides, because halides can interfere with iodine absorption in the body. Then there are the vitamin markers, which are your B vitamins via the branched chain catabolites. And I also check for methylation markers like folates and B12. B6 is also checked as well. And then biotin, because that's super important for trafficking oxalate out of the body. But in addition to that, I check to see if there's dysbiosis. A lot of times people have yeast overgrowth, they have mold overgrowth, and then they have other markers indicating that there is imbalances in the GI tract. And we talked about the importance of GI tract for the proper metabolism and removal of oxalates out of the body. Then, of course, there's the actual oxalate markers on the test. So we're talking about, and the organic acid does this one as well, talking about the actual oxalate markers. Of course, remember, it's urinary. It's not on a tissue level. So again, if you're not excreting it, don't assume you don't have this problem. And then there's glutathione, which is an antioxidant, oftentimes becomes very, very depleted. A lot of times that's due to the depletion of NADP and NADPH, but it could also be precursors to glutathione. It could be B6 deficiency, or it could just be the depletion of glutathione because there's just so much toxicity in the body. CoQ10 is an antioxidant that needs to be checked, and then there's a lipid peroxidation that's a consequence of oxidative stress, and then there's the DNA damage marker as well, which is really important to show you how much damage is happening inside of your body. And this marker is really important because this can be an early detector to see if there's potential for cancer in the body. And finally, my most favorite one is the Tree of Life genetics, showing all the different pathways that could be involved in the situation. So here's your oxalate genetics over here on the right. There's your PON1 for the glyphosate, which can contribute to oxalates. Here's your primary hyperoxaluria. And then we've got the KEAP1 and the NRF2. I found this to be very, very significant with people, especially with bladder pain syndrome, because of the role NRF2 has in the integrity of the bladder lining. So checking to see if that. Then there's iron overload. Surprisingly, I see a lot of people who've got iron overload, which is important information for you to have if you decide to embark on a carnivore diet. You have to understand that you're going to eat a lot of red meat. Is that going to be problematic if you have iron absorption or over absorption? And this can also, copper toxicity is also another thing that can be revealed, mainly from the hair tissue mineral analysis, but also the iron absorption markers can also help with understanding what copper is doing in the body as well. And then on here, there are some histamine pathways. I didn't show those, but they're also important. And then I didn't demonstrate that. Oh, there's also glutathione pathways as well, and also the Fenton reaction. So all very important because all that oxidative stress in the body can absolutely lead to increased oxalates in the body. And so once you've done the lab testing and we've gotten some information, there are some different things that we can do as interventions. Of course, the low oxalate diet is super helpful. But again, if you don't fix your biochemistry, if you don't fix your gut, I just don't think it's going to give you what you need to heal. Utilizing calcium therapeutically, magnesium, citrates if you tolerate it. B6, some people might need a higher dose, but again, with care. I think intermittent fasting is phenomenal for doing a lot of healing, but unfortunately, some people cannot tolerate that. Again, that's that disruption in gluconeogenesis. That needs to be fixed first biochemically. Vitamin B1 can be used very well therapeutically. MSM powder, Epsom salt baths, binders, and I should also mention Zeoboost as well, or Zeocharge. So that's the zeolites that I like to use now, and fulvic charge is fulvic acid. And then other things like Histoqual, Visbiome, and then different probiotics such as HG58 and Megaspore, and even the immunoglobulins can be very helpful. So here's where you can find me. If you want to work with me, I would love to meet you and see if I can help you. Even though my primary focus is on bladder pain, that's just kind of where I landed because of my own struggles with interstitial cystitis and bladder pain. I do take patients from all different backgrounds and all different health conditions. And if you have challenges with your gut health and some of the other things that I mentioned, like fatigue, brain fog, blood sugar dysregulation, you could be dealing with an oxalate problem. And I think it's really important for you to investigate to find out what's going on inside your body, so you can get to the road of healing, and so you don't have to suffer anymore. And that's really what the bottom line is. We want to have a better quality of life, and I'm here to help you. So I hope you enjoyed this presentation. I hope it was eye-opening and insightful for you. And out of everything, I really hope you learned something today. Thank you very much for being here. Bye-bye.